So hi everyone, and our group worked on DNA detectives, which is related to COVID-19. We have Sudharsan, Jeffrey, Justin, Ruta, and Candice. All right, so you guys go ahead if you're ready.
So hi, my name is Candice, and the name of our project is DNA Detectives. Next slide. So background and motivation. The reason we started this project is because SARS-CoV-2, the virus that caused COVID-19, is a RNA virus prone to mutation. We want to track different strains of the virus for containment and prevention. We can track features of each genome to predict where it originated from. Therefore, we created a model that can accurately predict the origins and evaluate mutations and variation of various SARS-CoV-2 samples.
So our data comprises different genome sequences of SARS-CoV-2. So we use the data to find mutations, and we often use the convention. So we often displayed 0 as no and 1 as yes. And we also figured out the number of sequences in 3 regions: Oceania, North America, and Asia. So the figure that you're seeing right here displays 2 sequences, which are really the last 2 rows, the 2 colored rows, each of SARS-CoV-2 and the bat virus known as RATG13. So if the 2 sequences have the same color at some location, it means that they have the same base at that location. And if the colors are different, then they have a different bases. And different bases means mutations. So using Python libraries such as matplotlib and pandas, we were able to create graphs and data tables like you can see right here. So the graph here describes the number of mutations to the number of sequences from any location in general. So as you can see, only a small fraction of all the sequences that we found in the world have severely many mutations. Like for example, over here towards the very right of the graph, there's about like between 350 and 400. But most of the sequences that we found do not have that many mutations. And the data table that you're seeing right here shows only a small portion of 5 sample sequences that we found. So those are labeled in these different rows right here. But this very first row, where it says like 1_A and then T, G, and A. So those are actually the bases of the sequences. So the 1s and 0s. So the 1s represent, the 1s basically mean that corresponding base is present, and 0 means that it's not present.
So a large part of our methodology was data preprocessing, and this is because some countries that we took data from had many more sequences of SARS-CoV-2 available for research. If we trained all the data as is, it would cause the model to underperform in predicting where a sequence came from if that sequence came from an underrepresented country. And so how we solve this is that we cut out the excess sequences from the countries that had a surplus of them. And we also converted countries to continents. We grouped them in continents to simplify the data and to group all the countries on the same continent with a low number of sequence strains. Another part of our methodology was the lasso penalty in cross-validation. So we use multinomial logistic regression to take a look at the sequence of different COVID strains and try to predict which country it came from. And so we used something called the lasso penalty, where we tried to address the fact that there were too many features we had to deal with. And so we tried to reduce them by using this lasso penalty. And so we can also reduce our loss this way. And so we have to optimize a number called lambda so that the loss will be as small as possible. And we use cross-validation to do so.
So the results. So below is a confusion matrix that shows the accuracy of our first logistic regression model, and it's around 91% for for our training testing data, and yeah. So for example, it predicted there were 27 genomes that came from Asia, and it predicted 26 of those 27. So these are the results from our second model, which used a lasso penalty reduced in which we reduced the data, used the lasso penalty, and also used cross-validation. And the result of these things allowed us to increase our accuracy and reduce overfitting.
So some things we can take away from this is that feature reduction is important, and this is because we can use specific features like biomarkers to determine the origin, as well as reduce overfitting to increase the accuracy of the model. And feature reduction will also lead to a decreased processing time. And cross-validation is also important because it can, but doesn't always improve the accuracy of the model. So what you do is you find the best lasso penalty, and then cross-validation can be implemented to train the model by splitting up the training data into a training and validation set, and the accuracy of the training sets is then evaluated using the validation set. And this can help us because we can use it to find the best average accuracy of the training data, but depending on the random nature of the data, it may not always be beneficial to the model. So some applications of this is that machine learning can help us observe mutations and the origin of not only the coronavirus, but in other diseases or bacterial infections too. And it can be applied to other diseases and viruses, as well as assist with finding new treatments and preventing the spread to contain it. But AI is not only applicable to the field of medicine. In fact, AI is impacting numerous industries like manufacturing to assist with quality control, in the automobile industry, like we can see with self-driving cars, and even the agriculture industry because of machines like AI bots, which can assist with harvesting and planting crops, like seen in the picture. Thank you for your time.